All right, we're here to talk about translation and go over the process of the conversion from the mRNA into the actual protein. So when we're talking about translation, this is a part of protein synthesis that is going to occur at the ribosome because the ribosomes are the organelle responsible for putting together the proteins. In prokaryotes, this is actually coupled with transcription since prokaryotes do not have the nuclear membrane similar to we do in eukaryotes. It's one of the things that separate us, a lack of a nucleus in prokaryotes. So as the prokaryotes are transcribing the DNA into mRNA, they can actually start the translation process as well. In eukaryotes, they're completely separate processes because the mRNA has to leave the nucleus to find the ribosome and get this going to make the proteins and put the amino acids together. Now, both of them involve the same kind of chemical code though. They're both read the same way and they both occur the same way, just a difference of having the nucleus versus not having the nucleus. So when we look at this picture, we see the red rectangle on the bottom. That shows where translation is occurring. Obviously, it's outside the nucleus at a ribosome. Ribosomes can be either free-floating or they can be attached to the rough endoplasmic reticulum. Just depends on the ribosome. So when we talk about amino acids, we have to know that mRNA has codons, and codons are always what's going to determine what amino acid is attached to the polypeptide chain. Every 3 bases on the mRNA is going to represent 1 codon, and 1 codon is going to equal 1 amino acid that's going to be added to the chain. And this code was broken in the early 60s, very shortly after Watson and Crick showed the structure of DNA. That sort of started this whole scientific discovery thing of how the cells actually work. So when we talk about codons, there's a couple codons we need to know about. The start codon is always going to be AUG. That's going to be the first one on the mRNA that's going to start the coding process. That's why it's called the start codon. And it's always going to be methionine. That doesn't mean methionine is always going to be the first amino acid in a chain of a protein, because sometimes proteins get modified, and we'll talk about that in a little bit. There are 3 different stop codons: UAA, UAG, and UGA. And when you look at our codons, obviously codons are 3 bases, and there's 4 possible bases. They could be uracil, adenine, guanine, or cytosine. If you have 4 times 4 times 4, that means that there are 64 possible combinations for codons. Since 3 of them are stop codons, that means 61 codons actually code for amino acids. Every amino acid has only one, or every codon, I'm sorry, has only 1 amino acid associated with it. But when we look at the codon chart on the next page, we're going to see that amino acids like leucine have 1, 2, 3, 4, 5, 6 different codons that code for that 1. That's because there's only 20 amino acids and 64 different combinations. So the codons have to match up with multiple amino acids. And you may see this codon chart in other forms. Obviously, on this one, you see the first letter of the codon is on the left, the second letter is across the top, and the third letter is across the right side. Sometimes you'll see it in a circular form where you have one big letter in the middle, smaller ones in the second ring, and even smaller ones in the third ring that go to different codons and amino acids. They all work the same way though, and if you look at them, you should be able to figure out how they work. The nice thing about codons and amino acids though, this is a universal code that is very consistent among all organisms. And anytime we see stuff that is universal among all organisms, that tells us that, or provides evidence that we had a common ancestor at one point in time. This is one of the big things biologists point to that say, hey, we had to have a common ancestor if we're all doing things kind of the same way. And that's one of these things that leads into the whole evolution talk that we'll talk about in about a month in class. When we look at the process of translation, it is very important that tRNA and mRNA match up correctly. If you remember, mRNA has codons. tRNA is going to match up with those codons, which are 3 bases long, with what's called an anticodon. The anticodon is going to be complementary to the codon, and each tRNA has 1 amino acid, but it may have multiple anticodons that are going to match up with it, because remember, we saw that there were multiple amino acids or multiple codons that went to each amino acid. So a tRNA can have multiple anticodons, but they always have to read the mRNA correctly, because the amino acid is always going to be determined by the mRNA codon, and that is very important information that people confuse quite often. When we look at tRNA, It has a 5' end and a 3' end because tRNA it's an RNA molecule. It is a nucleic acid. So it has all the bases just similar, and if you look at this picture on the bottom, you see the CCG acts as the anticodon that would match up with the GGC codon on a mRNA. There are not as many tRNA molecules as there are codons, and that's because you'll find multiple anticodons on each tRNA molecule. The ribosome is where all this magic happens. When we talk about a ribosome, there's two subunits. You have a large subunit and a small subunit. I don't care about the specific numbers, but that's why they're called. Small subunit only has one rRNA and about 33 proteins. Hopefully you remember rRNA is a ribonucleic acid or ribosomal RNA, and the large one has more of those and more proteins fit together. So it kind of looks like a weird shaped hot dog bun when we look at them together, and they fit together kind of like a puzzle with the mRNA running between the small and large subunit. When we look at the large subunit, you'll notice on this picture over on the right that there's 3 spaces, and each of those spaces fits 3 bases from the mRNA, which, remember, 3 bases of an mRNA is a codon. So the E, the P, and the A are all sites where codons reach. And if you look at the mRNA, you can see AUG isn't always the first 3 bases. It could be in the middle. That just tells the ribosome where to start the actual translation process. So the A site is the first place that the codon is going to move through. And you can see the mRNA enters the ribosome in the 5' end, and it moves down towards the 3' end through the ribosome. So the A site is the first one. That's the amino acid site. That's because what's called a charged tRNA is going to bring its anticodon and line up with the codon in the A site. A charged tRNA means that the tRNA has an amino acid attached to it. Once a charged tRNA gets to the right spot in the A slot, then the ribosome is going to move down the mRNA, or the mRNA is going to move through the ribosome, however you want to look at it. It doesn't matter. That codon will go to the P site. The P site is a polypeptide site, where that amino acid that's attached to that tRNA is going to get bonded to that polypeptide chain with a peptide bond. Once it bonds to the polypeptide chain, that tRNA is going to let go of the amino acid, and then it's going to go to the E site where the tRNA is uncharged now, and then as soon as the tRNA gets there and it's uncharged, it's going to leave the ribosome, and that's why we call the E site the exit site. So let's look at the steps of translation and see how all this stuff actually works. First, we got initiation. Well, in initiation, there's going to be a complex of a charged tRNA and a small subunit bound to the mRNA. You need to have those 3 things: the mRNA, the charged tRNA, and the small subunit all sort of put together. Once that happens, the small subunit is going to move along that mRNA until it finds that start codon, that AUG that codes for methionine. Once it finds methionine, it's going to move, and the large subunit is going to attach to it. Again, that methionine may get released later, doesn't matter, proteins always get modified. The large subunit is going to attach over there, and methionine will eventually be in the P site, because that's going to wait for the next codon to get matched up with the A site, and there's things called initiation factors that hold all this stuff together to get it going. So if we look at this picture, we see the small subunit and the mRNA attached there. The tRNA with the anticodon comes over and finds that AUG, and then the large subunit puts that AUG codon into the P site. So now we see a charged tRNA there, we see the methionine, that's what it's charging the tRNA, we see the small and the large subunit, and we see the ribosome. Next, the chain is going to get elongated. tRNA anticodons are going to join that mRNA codon, and they're going to start matching up. They're going to go into the A site first, and once they get into the A site, the amino acid is going to get attached to the amino acid in the P site, and it's going to keep going. After the tRNA in the P site releases a methionine, that tRNA is going to go to the E site. And I know this might be a little hard to follow because I'm talking about a very active process and we're looking at static pictures, but as you get going, we'll be all right. The tRNA that was in the A site then takes the place of the original one in the P site, and then a new one is going to move into the A site. So we have sort of this assembly line going. And as we look over here, we see that AUG is still in the P site at first, and we see the next anticodon in tRNA coming in with a charge that's charged with an amino acid. As soon as it gets into the A site, that amino acid from the P site attaches to, it looks like proline there, the PRO, it attaches to the proline through a peptide bond. And once it attaches, the tRNA with proline moves over to the P site. That initial tRNA moved out of the E site, and now you have an open A site. And that's going to allow more tRNAs to cruise through. Now this process keeps going until you get to a stop codon. And once you hit the stop codon, and that's going to enter the A site on that large subunit, that's going to tell release factors to come into the A site and release the large subunit from the small subunit, release the tRNA, and release the polypeptide chain. And everything's just going to sort of go away. So when we look here, you can see the little chain of amino acids that are put together. That's our polypeptide. We see they're attached to a tRNA. And then we see the UAA. That was one of those 3 stop codons. That tells release factor to come in. As release factor comes into the A site, that tells everything to sort of break apart. And that terminates the actual translation on that ribosome. Now, one of the cool things our cells can do is they can create something called polysomes, where multiple ribosomes will actually attach to the same mRNA at the same time and start making the same protein over and over and over. So this is a really quick way to get a lot of protein going through, and this happens quite often in our cells. That's the picture on the far right shows what it really looks like under an electron microscope. And you can see, if you look at the top of the cell, you can see the growing polypeptides are a lot shorter than when you look at the bottom of the mRNA strand. You can see how many ribosomes you got there. I mean, there's about 15 of them or so going on there, creating multiple proteins, because proteins do everything for our body and for our cells. So we need a lot of those things sometimes. So these polysomes is a really quick way to make a lot of protein off of that one mRNA strand. Now, once the proteins are made, it's not done. That protein is going to get modified, and a lot of times when translation's occurring, maybe that protein's not even going to get used in the cell. If it's something like a hormone, it's got to get transferred through the one of the ways that proteins can be modified is through something called proteolysis, which, if you look at the words, proteo refers to protein, and lysis means to break up or to burst or to cut. Well, endoplasmic reticulum is one of those organelles that's responsible for modifying proteins. So inside the endoplasmic reticulum, that is going to cut the proteins up and make them into smaller proteins. And an example that the book gives us is something that HIV does. HIV is human immunodeficiency virus, and it's going to use something called proteases to cut viral polyproteins and make more little HIV viruses. Well, some of the drugs that we use inhibit this protease from the HIV and help prevent that spread of the HIV virus throughout the body and throughout other cells and to other people. Another way that we can modify protein is through glycosylation, which hopefully if you hear glyco, you should think sugar. And a glycosylation is when you form glycoproteins, which we talked about a long time ago when we were talking about cell membrane stuff. Glycoproteins were on the outside of that cell membrane and helped with receptor signals for the cells so that things could get in and out of them. Well, basically, sugars get attached to the protein, and they'll help stabilize proteins, they'll help stabilize the extracellular matrix and stabilize cells within the body. The last thing that can happen to proteins is called phosphorylation, which hopefully you realize that means you add phosphate groups to the protein. And protein kinases help with the creation of these phosphoproteins, and they are really important in cell signaling and sending messages from cell to cell. So when we look at translation, we can see that we got a ribosome translating a protein in the cell somewhere. We got that mRNA strand, we got this long polypeptide chain. Well, that polypeptide chain can go one of 3 directions. It can get cut up into shorter chains, it can add some carbohydrates there and those little pink hexagons that are attached to it, or it can add some phosphates, depending on which way the protein's modified. And when we look at how the proteins are used, they can be used in a variety of ways. They may be used inside the cell, they may be used outside the cell, and hopefully you remember ways things get in and out of the cell. You can see that the protein gets modified in the rough endoplasmic reticulum. The rough endoplasmic reticulum is passing it off to the Golgi apparatus on the right side of this picture. The Golgi apparatus modifies it even more and packages it up to send it either out of the cell through exocytosis, or maybe it creates a lysosome that'll help digest something. Maybe the protein goes into another organelle within the cell. There's a wide variety of things, because remember, proteins do pretty much everything for us. And this whole process of transcription translation is what we call in biology the central dogma, because this is kind of the basis of how cells work. And this is a very big process. Hope you enjoyed this. Hope it wasn't too much for you. We'll be talking about this in class. I will see you soon.